Hello everyone, I'm Sarah Swinton, and today in this video we are going over the history towards evolution. Now if we look at the word evolution, it has the word evolve in it. Now evolve actually means to change, so it's a change in a population over a period of time. So, when we're looking at the history towards evolution, we're actually looking at the origins of life or the beginning. So, from our beginning, we're actually going way, way back in time from the 1600s to the 1800s, and we're going to start there. So, prior to the 1800s, it was widely thought that organisms could only arise from non living matter. That was known as spontaneous generation. So, living things came from non living. Now, this is also called abiogenesis. So, if you look at the word, a means no or not, bio, life, and genesis, the beginning of. So, it's the beginning of life from non living things. Now, really cool fun fact, it was actually Aristotle that was the one who proposed the idea of spontaneous generation, where living things came from non-living things. Since he was so renowned for all of his work in the scientific field, the Catholic Church 100% backed him up. Because of this, the Catholic Church actually shut down many of the theories against Aristotle's abiogenesis until after 1859, which is absolutely crazy. So the first person to go against Aristotle's idea of spontaneous generation was Francesco Redi in 1665. So he designed an experiment to disprove of an idea that maggots could grow from rotting meat. So abiogenesis. He thought that maggots had to come from flies, which laid eggs in the rotten meat. So in his experiment, he had meat that was exposed to the flies, and of course they grew maggots. The next thing he did is he sealed the container and he wanted to see if there would be any maggot growth. Well, since he sealed the container, no flies could go in, there were no maggots. So he's like, hey, by the way, um, maggots cannot just grow from rotting meat, they had to be carried in by the flies. But there's a lot of backlash. So again, the Catholic Church was like, hey, no, there's no vital air, so that means there is no growth to occur because you need air in order for living things to grow. So then he designed the other part of the experiment where he covered his container with gauze so that way air can get in, but no maggots could actually, or flies could actually go inside. Because no flies could go inside, there were no maggots. So here he was able to conclude that maggots had to come from the flies and they did not actually grow from the rotting meat. So from here, he was able to conclude that biogenesis was correct, where living things had to come from living things. But again, this is the time period where we were very much against this, so we needed more evidence to show that biogenesis is actually true and not spontaneous generation. So this brings us to Lazzaro Spallanzani in 1768. So quite a few years later, he designed an experiment again to disprove of the idea of abiogenesis. So he wanted to disprove the idea of microorganisms, a lot of your bacteria and stuff, just appearing from broth that's left out. So he believed that microorganisms were actually carried by the air. So what he did is he sterilized the broth first by killing any possible microorganism growth. When he did so, he left the broth overnight, exposed to the air, and again, over time, the microorganisms end up growing and you had the growth. So then he wanted to see what would happen if he covered it up. So he repeated the same experiment, but he sealed the container, and after multiple days of no exposure to air, there was no growth in the end. Again, we had a problem. There's a lot of backlash. Again, this is the time where Aristotle's beliefs were very high up there. So, air was considered to be vital for growth, which means that this could not actually be true. But according to Spallanzani, in his conclusion, microorganisms were carried in the air and they did not just grow from the broth. So this brings us to Louis Pasteur in 1859. So many years later, Pasteur actually redesigned Spallanzani's experiment to disprove of microorganisms actually growing in the broth. So he also believed that the air was the one that actually carried the microorganisms. So again, what he did is he designed a flask with a S-shaped neck that was designed to keep the dust out, but air could actually go in. He sterilized the broth just like Spallanzani did to kill any type of microorganisms that were already present. He let the broth sit for actually months, and after months, there was no microorganism growth in the broth. Then what he did is he mixed the broth with the dust that was trapped in the neck, and after 1 day, microorganisms were growing in the broth. So from here, abiogenesis was officially disproved, and we moved from abiogenesis to biogenesis. So, bio life, genesis beginning of. So, the beginning of life, life had to come from life. All right, so now that we know that living comes from living things, this brings us to where did the first living thing come from? So, our first organic compounds on Earth may have actually formed under high energy conditions like high UV and high lightning. And our early Earth's atmosphere would have had very little or no oxygen. Now, remember, in order for things to be living, we have 6 essential elements. Those 6 essential elements are your NC hops or your chinops or nchops. So you need nitrogen, carbon, hydrogen, oxygen, phosphorus, and sulfur. Those are your 6 essential elements to make up your macromolecules of carbohydrates, proteins, lipids, and nucleic acids. So this is actually going to bring us to experiments from Miller and Urey. So Miller-Urey's experiment was actually designed to disprove of the possibility that macromolecules, carbohydrates, proteins, lipids, and nucleic acids, could just actually be formed in early Earth's atmosphere. So they were getting rid of this idea. So they created their experiment, and basically, first step, they mimicked Earth's atmosphere by mixing water vapor, methane, ammonia, hydrogen gas, carbon dioxide, nitrogen gas, and so much more. In the second step they did is they took electrodes and they created an electrical spark. This would mimic lightning for the chemical reactions to actually take place in the atmosphere. What they also did to mimic early Earth's atmosphere is they cooled those gases with circulating water or the water vapor that would be in the atmosphere. And in the end, they actually took a sample and they found organic compounds such as the monomers of proteins, the monomers of nucleic acids, the monomers of lipids, and the monomers of carbohydrates. So he was actually able to show that, yeah, actually, it is possible. So you couldn't actually disprove of the idea. Well, this had to bring us to our first actual organism, or your prokaryotes. So this is the endosymbiotic theory. So remember, endo means inside, sim means together, and bio life. So basically, organisms came together to help each other out by engulfing another organism, and that's how you get your first cells, pretty much. So, early Earth's atmosphere would have supported the formation of anaerobic prokaryotes. Remember, A or and then aerobic, so no oxygen would actually be present. So, prokaryotes that don't need oxygen. Now, an increase in UV in the atmosphere would have actually supported the formation of some of our first photosynthetic prokaryotes. Now, this actually probably looks very familiar. This looks very similar to a chloroplast. So, photosynthetic prokaryotes look very similar to modern day chloroplasts. Well, an increase in oxygen in our atmosphere could have actually supported the formation of aerobic prokaryotes. Now, you guys might actually look at this and go, hey, this might look familiar. It should. It looks very similar to a mitochondria. So, aerobic prokaryotes actually look very similar to modern day mitochondria today. So this brings us to the actual endosymbiotic theory. So a prokaryote could have engulfed another prokaryote to form some sort of symbiotic relationship to form our first eukaryote. So prokaryote, no nucleus, eukaryote with a nucleus. So our first eukaryotic cell could literally be a bacteria cell that engulfed other bacteria. So then we could have ended up with a ancestral eukaryote just like this. So this is called endosymbiosis or the endosymbiotic theory. Endo means inside, symbiosis, it's a relationship between organisms. Now we're actually going to lead towards natural selection. So we're actually going all the way back to Aristotle again. Now Aristotle had some great ideas. However, what we do know now is a lot of his ideas were actually pretty flawed, especially when it came to biology. But going back to his ideas, Aristotle actually believed that species were fixed creatures and they were arranged by their complexity. So, because of this, he came up with his simple classification system. His classification system is that all living organisms were either a plant or an animal. That was it. Those are the only two categories. There was no room for protists, fungus, bacteria. It's just plants and animals. So this actually brings us to Carolus Linnaeus. Now, Linnaeus, he did not actually believe that species were fixed creatures. He thought that things could actually change. So he did come up with a classification system that allowed those different changes with any new information that we find. Science is not fixed. Science is constantly changing. We're constantly rearranging things whenever we come up with new evidence. That's the beauty of science. So because of this, he also assigned organisms by with Latin names, which we actually call binomial nomenclature. So bi means two, nomial name, nomen name, and clature system. So binomial nomenclature, it's a two-name naming system that every organism actually has, and that way, if we find new species, we can already give it a name based off of this classification system. This classification system is what we use today. Now, Charles Lyell, he came up with a theory of uniformitarianism. Basically, this is where geological processes are going to occur at uniform rates. So, his principles of geology he published actually explained that geological processes shape the earth as we have it today. So, basically, the sediments of rock and stuff like that is going to occur uniform. But this brings us to Jean-Baptiste Lamarck. Now, Lamarck was actually the first scientist that understood that changes in populations can actually occur over time. His ideas, however, as amazing as it was, very flawed. It was very flawed. He stated that changes are adaptations to the environment that were acquired in that organism's lifetime. That's not actually true. He had the right idea, he was off. So this is where he actually missed the mark. So his idea was that if we have a short-necked giraffe, now he did a lot of his experiments with giraffes or his ideas, because he found there were short-necked giraffe fossils and then there's long-necked giraffes. So his idea was that if a short-necked giraffe couldn't reach the food, he would actually acquire the characteristics for the offspring over time. That's not exactly how it works. How it would really work is if you have a short-necked giraffe and they can't reach the food, they die, which means any organism that was able to reach the food with longer necks, they would survive, pass those traits on. That would be how it actually work. Um, so yeah. His hypothesis is use and disuse, where organisms could either change the traits by using or not using them. Again, he missed the mark here. But again, this is giving us great ideas for actual natural selection. So Thomas Malthus, he hypothesized that population size was actually limited by resources such as a food supply. So populations are fighting for survival by the limited resources. And now we get Charles Darwin. So this brings us to actual natural selection with Charles Darwin. So with Charles Darwin, he sailed on the HMS Beagle from England for a detailed hydrographic survey around the coast of South America. And this is his route that he took. So he was able to discover Galapagos Islands, where he observed giant tortoises and their traits. He also observed finches on the island. And he also noticed that a lot of the finches actually resembled other finches nearby. So he thought that there was actually a common ancestor from the mainland finch, based off the different species that he found. So he saw patterns of diversity. Individuals of a population varied. There were no two organisms that were exactly alike, which is so cool. Genetics are awesome. So that means the variations or genetics are inherited. All right, so looking more into Darwin's finches, again, the idea of a common ancestor is we had our different Galapagos Islands, and each island had different food sources, which will give you different species of finches. So if the food source is seeds, the beaks that he would find were thick and stout. If the main food source were insects, then the beaks he found were curved and sharp. If the main food source were grubs, then the beaks then were thin and sharp, and that's what gave them the different species or speciation. Now, descent with modification, this is the idea that species changed over time, and that gave rise to new species, and they shared that common ancestor. Now, maybe this thing looks familiar, at least I sure hope it does. So this brings us to different patterns of evolution with divergent evolution as our first one. So divergent, di means two. So this is splitting from a parent species to a new species. We call that speciation. So if, again, you have your mainland finch, looks like that, and then you have your different species of finch, still finches, you got your goldfinch and your greenfinch. And there are many different types of finches out there, but this is an example of divergent evolution splitting into two. So then convergent evolution, remember co means together. So convergent evolution, two parent species become increasingly similar due to this similar environments. So if you have a dolphin and a shark, two completely different species, they have similar traits or similar features like fins and a tail because of the environment they live in. And then coevolution, again, co together, so different species rely on each other for survival. So predator prey relationships are a great example of coevolution. You have your predator, you have your prey, and basically anything that affects one population will affect the other population in the end. So again, speciation, this is the production of a new species of organisms, and this can be resulted from a geographic or reproductive barriers. So a geographic isolation is when a population gets separated by a geographic barrier or an actual barrier in their environment. So then each new population will then have a different gene pool, which leads to a new species that's formed. So example is this. You got your green beetles, you have your brown beetles, some sort of geographic barrier separates them. Now you have a population of just green beetles and a population of just brown. So the beetle population got separated and the gene pool changed to the new populations, and that leads to speciation. The other type of isolation is reproductive isolation. So the population will separate themselves with preferential mating, where speciation then can occur. So reproductive isolation, you got your green beetles, you got your brown beetles. Your green beetles mate with only green, your brown only mate with brown, and then they end up splitting themselves up or segregating themselves, and they isolate themselves reproductively. So then again, descent with modification, species today do look different from the ancestors or from all the different fossils. So each species then had to descend with changes from other species over a period of time, and that's what you can actually see here. So you got your common ancestor, your original horse-like species, and then you have your modern horse here.